Matute, and I come from the University of North Carolina. And I'm going to talk about behavioral defects in hybrids of Drosophila. We have had a beautiful section actually that has studied, that has gone after the genetic basis of reproductive isolation, that has revealed essentially how sexes interact in animals and how actually this leads to the preclusion, like essentially to gene flow that occur between species. Reproductive isolation is one of the central tenets of the studies of speciation. And the reason for this is that actually there are several, and one of them is that it involves a particular trait with a particular process that actually precludes gene exchange. Depending on what is there, the traits that actually preclude such gene exchange occur in the reproductive cycle of an organism, they can be divided into three particular traits. These divisions are rather arbitrary, and you can actually divide them in however you want. Pre-mating isolation occurs if, as the name implies, before the mating occurs. Everything that is involved in ecological isolation, mating behavior, traits like that. Gametic isolation is all those interactions between gametes and female reproductive tracts, for example, that actually go awry and just don't allow the zygote to get formed. And post-zygotic isolation, that is going to be the focus of this talk, occurs when a hybrid actually gets formed, but it shows any kind of like fitness defects. Drosophila has been one of the main focus and one of the powerhouses to try to understand patterns of genetic isolation, how genetic isolation and post-zygotic isolation are correlated, but also to try to understand the genetic basis of the trait. There are two particular traits that have been studied under this umbrella of phenotypes: hybrid viability and hybrid sterility. I'm not going to be talking about those today. In this particular case, I am showing Drosophila melanogaster and Drosophila similans. This particular cross has been absolutely instrumental, and some of the giants of the field actually work on this and have been trying and succeeding at isolating alleles involving hybrid viability and sterility. You will hear names as big as David Presgraves, Daniel Barbash, leading families that actually have been going and doing extremely fine genetic work and trying to identify these genes. One of the lines that we have in the lab that is also another phenotype that causes reproductive isolation is hybrid sterility, for example. In this particular case, I am showing you the testes of of a group of species that is called Drosophila jacobacate. And you can see that the pure species are, like the testes of the pure species have a sperm, and every other possible cross is a completely empty testis, precluding the possibility of these individuals producing progeny. As I was mentioning before, Drosophila hybrids can be produced in many different combinations, and for example, in this particular case, I am showing how to make a melanogaster female similans cross that produces F1 females that, even though are viable, are completely sterile. The males die as larvae, and this has essentially just generated a powerhouse of genetics and trying to understand the genetic basis. I got particularly curious after working with the tremendously talented Grace Lee on essentially what kind of reproductive isolation and what kind of defects these hybrids might also show besides the sterility or inviability. She actually generated a very clever device into how to find out whether a single new allele and a new gene could lead to the preference of different foods in Drosophila mauritiana. That made us wonder whether actually hybrids that are produced in the lab can find food with the very same scheme that actually she designed. The design is pretty simple, and it's essentially a wine maze in which you actually put the starving flies. You start them for 12 hours, and at that point they will just go for whatever food they find, if they're all right, and put two different vials, one that is completely empty and one that has a source of food. We had three hypotheses. The first one was that there was not going to be any sort of differential distribution between the three vials. They were going to go randomly, and they were going to just assort themselves in random places. The second possibility is that actually individuals were going to find food. And the final possibility is that they were going to be repelled by food. This is a very stupid decision, of course, because they're going to die. The first control that we did and the first experiment that we needed to do was to find out whether actually pure species can find food. That's the first proof of principle that we needed to do. And what I'm showing you here is the pair that I already talked about that is essentially one of the main focus of my lab. that is Drosophila jacoba and Drosophila santomea. When you put individuals of the two pure species into this particular white maze, almost unavoidably they are going to find food, regardless of whatever the sex is. The story for hybrids is actually different. Here I am showing you the two colors are showing the two reciprocal crosses because the two females can be crossed with the two males and actually the two types of hybrids. And you can see that the difference between the female hybrids and the female pure species is absolutely staggering. All that is also true for individuals that are males. One of the things that you also can see is that these females that are hybrids are more likely to find food than male hybrids from the same cross. These are brothers and sisters. There is really no major genetic difference between them, besides the fact that some of them are females and some of them are males. When we went to a rather divergent cross, because this cross, the divergence between the two species is about 1 million years. When we went and repeated the experiment with a very divergent cross, that is a cross that we recently identified that can be done in the lab, that is Drosophila melanogaster to Drosophila tessieri, we found the same pattern. The hybrids actually are less good than the pure species at finding food, and that's when they understatement, as you can see here. There's a reduction of about 75% in females and about 90% in males. But also, the defect is also more pronounced than in actually the crosses that are more recently related. Here I'm showing you just the two crosses that I showed in the last two slides. But I am actually, what the message that I am trying to convey is that females between recently divergent parental species are more likely than hybrids between really divergent species. So there seems to be some effect of the divergence between the parental lines. However, this is 2 points, and with 2 points, we can do little. And both of these crosses belong to the melanogaster subgroup of species. The melanogaster subgroup of species has been a major focus of research. Pretty much every Drosophila biologist has had that phase, and has gone through the melanogaster group, including myself. And I am unlikely to be cured of it. However, melanogaster is only one piece into the giant variation that Drosophila is. We're talking about thousands of species here. And I'm just showing this phylogeny that we did a few years ago, just to show that this is a very, very reduced percentage of the variation. What we did next was to try to expand the results that we observed in the melanogaster group to as many variations as we could in the Drosophila genus. And for the most part, we succeeded with the exception of the Hawaiian species, which I just not worth it to be raised in the lab. Seriously. So what we did was to try, and we got hybrids from 12 different variations, 106 variation of 960 experimental blocks, and we measure the same trait. Very simple. Putting flies in the middle and see how they arrange themselves between having a choice of food and an empty vial where they unavoidably die. And we regress those results to genetic distance. This was the slide that I was going to present. And I gave my practice talk to my lab. And this is one of the huge advantages of recovery, like actually being able to recruit people that are just way smarter than you are. I got a school, and I was told that I should not show this, but I am just still giving it here. So you can actually see that there is a very strong correlation between genetic distance, negative correlation between genetic distance and the proportion of flies that actually find food, for both for males and females. However, this data set as it is presented has a series of problems. There is no phylogenetic correction, there is no replication because several lines have been used several times. For example, since I use melanogaster so much, I use it for several crosses, etc., etc. My lab actually helped me correct this. And what you have here is essentially the same plot, but instead what we are doing here is actually showing just genetic distance, the proportion of flies that find food. But every time that actually I use one species, for example melanogaster, I don't use it again. So essentially it corrects for that several replication of using one species into multiple hybrids. And what you can see here is that there is indeed a decrease into the ability of hybrids to find food with genetic distance. But you can also find that it's not a linear decrease, that it asymptotes around 20%. You might be wondering about what kind of defects are actually these hybrids that actually don't allow them to find food. And there are two possibilities. The first one is that they are going to the empty vial, or the second one is that they are actually remaining into the vial that they were put initially, and they are just not moving. And I have to apologize, I just realized that there may be colorblind people here, and I really deeply apologize. What you have here is the proportion of flies that actually stay in the, if it is red, they actually go to the food. If it is green, the ones that actually stay in the center, and if it is blue, the ones that are actually going to the empty vial. What you can see here in the red, the ones that actually find food decreases. But the two types of defects, the ones that actually, the flies that actually go to the empty vial or the ones that stay in the center vial, both of them are increasing with genetic distance. It seems to be a combination of two things. Flies actually going to the wrong vial, making the wrong decision, and flies staying completely still and simply preferring to die than actually. Moreover, we did this experiment for those species that we could produce females and males. And what I'm showing you here is essentially the same genetic relationship that I showed you before between genetic distance and proportion of flies that find food for females and males. What we find is that there is a strong effect of male, of the sex, and males are consistently worse than their sister at finding food. This is consistent with Haldane's rule. It's not Haldane's rule because Haldane's rule is specifically formulated for intrinsic post-zygotic isolation in the form of inviability or sterility, but it's consistent with that pattern, in which males, or in this, well, more generally, individuals of the heterogametic sex, males in Drosophila do worse than females when there is a particular trait that is involved in reproductive isolation. Let me wrap up, because we are ready to go to lunch. What is happening here is that I am showing you some really old school and basic experiment in which we are detecting that hybrids are not as good as the pure species at finding food. This is a very basic behavior. I know that this is an artificial setting, but we are giving hybrids the possibility of finding food or not finding, and consistently dying. And some of the hybrids just don't have the ability to find out that kind of resources. The second conclusion is that there is, that hybrids that are males are worse than their sisters. So there might be some effect of the X chromosome in this particular trait, and we are actually currently mapping whether that is the case or not. Finally, is that the behavioral defects accumulate with genetic distance, but that accumulation is not linear. There is a point that there is an asymptote that you cannot get worse than finding food than 20%. That is some other day. Finally, I would like to acknowledge to people that actually helped me quite a bit with this work. Jeffrey was a former technician of the lab that went to greener pastures in medical school. And David Turrisini and Aaron Camon that assessed me in the statistics of this and are much better than I am. Thank you very much.
Have you looked at temperature effects?
No, we have not. And actually, I have not thought about that until now, but it sounds very interesting.
You found the morphological effects between hybrids and flower beetles. Temperature makes a big difference. So.
Yeah, it would be really interesting to actually run these kind of assays at different temperatures and see. That is a cool idea. We're going to do it.
So you might have said this and I dismissed it, but my impression is that some of those fruit flies like fruit, some of them like vegetables, some of them like fungi, like mushrooms. Do you control for that sometimes?
That is an excellent point. Some of the species that we are using are specialists and some of them are generalists. The specialists, for example, Drosophila seychellia is restricted to Morinda citrifolia, or Drosophila desirae is restricted to Carinaria. For what we find here is that we didn't control for particular substrates, but individuals even from those specialists, like Seychellia or desirae, go to the fig. Because even though it's not their preferred source of food, at that point, they are even looking for any substrate or any community to survive. Yep.
So do you have a sense that there's a carryover effect between larval foraging and adult foraging? Did you look at larvae at all?
No, we didn't. But that's a really interesting question. We haven't actually worked with hybrid larvae for the most part.
Do you think that selection, in other words, do you think that there might have been larval mortality that might account for that 20% plateau, that the ones that are really bad at foraging all starve to death in the larval phase? Or?
I don't know. I don't know. I need to think a little bit more about that, but that's actually an interesting possibility. I am not sure what kind of genetic correlations exist between adult foraging and larval foraging, but that doesn't mean that it has not been explored. It just means that I don't know.
So given the genetic distance used and the complexity of the behavior, a lot of the complexities, let's say,
Well, that's a good point. We don't know. I think that this is a result of recycling practice indeed. I think that is going to be highly political. And it's likely to follow something like introduction as community compatibility. We don't know yet.
How does sterility play a role in it? So do sexually active females, are they more efficient in identifying food versus sterile females?
That is an excellent question, and I don't know the answer because it's going to be these two traits combined. So really, like virgin females are likely to not find food, but they're also likely to be sterile, more likely to be sterile. So I don't know how to differentiate this.